Hello, my name is Abigail Marciniak, and I'm excited to present to you my project, an oligonucleotide-based conjugation method for artificial antigen-presenting cells validating three-ligand attachment. In spring of 2023, my father was diagnosed with hepatocellular carcinoma, a very deadly form of liver cancer with a low 5-year overall survival rate. Thankfully, his cancer was early on, and the tumor was able to be removed, and he made a full recovery, which is unfortunately not something that can be said about many cancer patients. His ordeal inspired me to pursue biomedical engineering and piqued my interest in cancer immunotherapy. Unlike traditional therapies such as chemotherapy and radiation, cancer immunotherapy harnesses the body's own immune cells to target and kill cancer cells. In your body, there are immune cells known as dendritic cells that engulf pathogens and present their identifying molecules or antigens on their surface. With a variety of what we call stimulatory and co-stimulatory molecules, this dendritic cell will interface with a cell known as a T cell, and the T cell will expand in number and also be activated and know which cells to target after interacting with the dendritic cell. One way to go about cancer immunotherapy is to produce artificial antigen-presenting cells via droplet microfluidics. Essentially, we create something with a watery interior and a phospholipid bilayer that mimics a real cell. We then use something known as the oligonucleotide conjugation method, which is highly novel, and we use short base pair DNA segments, complementary strands that allow us to attach the ligand or co-stimulatory molecule we're interested in. The way this works is we have one strand with cholesterol, which embeds in the AAPC membrane, and another strand which contains the molecule we are interested in. My aim is to ensure that we can actually attach all three stimulatory molecules necessary. Currently, there is no known figure on what the optimal ratio of these ligands are, so we need to make sure we do thorough experimentation. One way we can ensure that it's possible to conjugate the ligands we want is through fluorescence and fluorescence microscopy. Essentially, we take one oligonucleotide that embeds in the membrane and a second complementary strand with a fluorescent molecule, and we incubate them together. We then verify via fluorescence if it attaches. If the experimental group shows fluorescence and the negative control does not, we can conclude that it is possible to conjugate the three co-stimulatory molecules we are using. As a researcher, I have learned many skills, including fluorescence microscopy, double emulsion droplet generation, as well as dealing with oligonucleotides in various solutions. Optimizing this ligand ratio and verifying this attachment will allow us to greatly improve AAPC therapy, which will improve the lives of many patients.